Hello everyone, I am Mohan and today I welcome you to my presentation which is about a collaborative research titled Massively Parallel Characterization of Transcriptional Regulatory Elements in Three Diverse Human Cell Types. The need for untangling the regulatory code in humans as of today has resulted in a global catalog of millions of regulatory elements. This has become a daunting challenge as we do not have a functional validation or readout which tells us if these candidate regulatory elements are active in each cell type. To address this, there exist high-throughput technologies such as massively parallel reporter assays or MPRAs, which enable for each region being tested its ability to increase or decrease the expression of a reporter gene as shown in green in this figure. The degree of enhancer-mediated activation is then quantified by the abundance of the reported transcripts. Keeping the daunting challenge in mind, we raise two important overarching questions. Can we scale this MPRA to a genome-wide level and test all the promoters and putative enhancers in multiple cell types? And can we develop a machine learning model with better prediction and interpretation towards understanding cell type specificity? We used our previously developed lentiviral-based MPRA framework or lenti-MPRA, which unlike other MPRAs provides an in-genome readout. As we go through our schematic strategy, we started by including thousands of candidate regulatory elements in both forward and reverse orientation. The elements were inserted in a lentiviral construct with barcodes, which in this setting from now on will represent the abundance of the transcript. The libraries are then infected into three diverse human cell types, and finally the integrated DNA and the transcribed RNA barcodes are sequenced to quantify the element activity score in terms of log2 RNA to DNA ratio. Upon completion of our lenti-MPRA in HepG2 cells, we observed that the distribution of element activity score in y-axis was strongly divergent between positive and negative controls, with promoters and putative enhancers spanning the range in between both extrema. And this trend was also observed in K562 and WTC11 as shown in this figure, thereby confirming that our scalable lenti-MPRA technique works. And now it's time for further analysis. One of our aim was to evaluate the relationship between promoter activity as measured by the lenti-MPRA technique and the gene expression of endogenous gene located in close proximity, which is then measured by the bulk RNA-seq. And to our surprise, despite relating to a very short promoter definition, the 200 base pair core promoters strongly recapitulated endogenous gene expression levels with 57% Pearson correlation in HepG2, and we observed similar percentage in other two cell types as well. These findings are consistent with prior models which showed that the core promoters centered at the TSS possess weak cell type specificity, are information dense, and could strongly predict gene expression levels. Now with our sizable MPRA, we aim to build predictive models of different architectures such as lasso regression, which uses biochemical features such as DNA-seq, ChIP-seq, histone ChIP-seq to predict element activity score. To determine the degree to which a model based on sequence features could predict the activity, we built MPRA-NN, a convolutional neural network which takes in the element region as one hot-coded sequence. To make the results directly comparable to the lasso regression model, we trained MPRA-NN using the same tenfold cross-validation on identical tenfolds of data as before used in lasso regression. The final prediction was then made on the held-out validation set. We also added a previously published large deep neural network called Informer into our pool of models, which is based on a CNN transformer architecture to generate a feature set of 5,313 biochemical predictions, which we further tunneled into another lasso regression model in order to predict the element activity. Upon benchmarking, we observed that the CNN-based MPRA-NN was able to outperform the lasso regression model, and the Informer MPRA outperformed all the other. Combining all folds of data, Our best model and former MPRA achieved a performance of a Pearson correlation of 83%, thereby confirming sequence models' ability to predict better than the biochemical models. Next, we took our MPRA-NN model for model interpretation and generated in silico mutagenesis predictions to use with TF Modisco Lite, which is a tool developed by the Kundajia lab to identify motifs for ISM predictions which had a higher effect. This strategy identified transcription factor binding sites which are cell type specific as mentioned in bold, and also identified transcription factor binding sites for a large suite of housekeeping transcription factors which are predicted to activate transcription and found in all cell types. We also discovered a motif of REST, which is a known transcriptional repressor and was only found in HepG2 and K562 cells. Next, we sought to validate the accuracy of the MPRA-NN's generated ISM scores for known enhancers and promoters. Here I show you one example, which is SORT1, for which we had previously applied MPRA saturation mutagenesis to measure the effect sizes of all possible variant effects in HepG2. Comparing MPRA-NN predictions for SORT1 enhancer to the MPRA data revealed that most of the relevant transcription factor binding sites could be detected, although the predicted effect sizes were exaggerated for other transcription factor binding sites. Collectively, we observed that a correlation of 47% for SORT1, confirming that the MPRA-NN, despite being trained on just the element activity score, could partially model the regulatory effects of individual genetic variants. In conclusion, we were able to scale our lenti-MPRA approach to test more than 680,000 transcription regulatory elements among 3 cell types, out of which we identified 40% of them being functional enhancers and more than 50% being functional promoters in all 3 cell types. We also observed that the human 200 nucleotide core promoter could strongly explain endogenous gene expression levels. And in our machine learning endeavors, we observed that the sequence-based models could outperform the biochemical-based ones, at least in our setup, and we could interpret our sequence-based models further to find specific transcription factor binding sites which modulate gene expression. Further investigation on cell type specificity along with the MPRA-NN code are available in our preprint and also in the QR code shown here. And in the end, my hearty thanks goes to Vikram and Fumitaka for giving me this opportunity to be part of this research and to all others who made this work possible. Thank you for listening.